Hi everyone, this is Karen. Today I'll be taking you around the lab. I'm a master's student and I'm currently studying plant sciences. I'm doing this super exciting project where I am studying an interaction between fungi that's inside the plant. In this video, you'll be able to see me running around the lab like I do most of the time. It was an exciting week. Lots of things happened. I tried filming using my new camera. Hope you enjoy the video. Monday, starting of the week, I managed to get up early. I was reading some papers. I usually go swim. Then I get to the lab at about 8:10. Today, I didn't have much going on in the morning. Before I started an experiment, I wanted to discuss some things with my day-to-day supervisor. So I was mostly working on the presentation for the lab meeting on Thursday. I haven't made that much progress yet, but hopefully it's gonna get there. Preparing liquid cultures. I had some bacteria. I want them to grow in liquid and extract their DNA. So I'm putting some bacteria in liquid that has nutrients and leaving these tubes overnight so that the bacteria grows tomorrow. Chemistry room. I literally live here at this point, but I never see anyone else coming in. I usually just bring my laptop here and work on things while I do my washing steps. 5 minutes is a bit too long to do nothing. I started running some protein gels. I was initially running 2 gels, but then one of them failed. I was thinking of doing another one, but then, nah, let's just leave it till tomorrow. I had a meeting with my day-to-day supervisor discussing some gels that did not go well. I still need to present it. It's not what I want to present, but things don't necessarily go the way you expect. While that's going on, here are my plants, which have been growing on nutrient jelly thing. Right now, they seem to be growing well, which I'm really happy. For now, I'm just removing water because they could contaminate the plates. I don't have enough time to repeat this experiment, and we don't want any contamination. Plants take weeks to grow. It's not like working with bacteria which grow in days and even hours. So that's why I can't mess this up. I love walking in here every single day. The building is so beautiful. The architecture is amazing. Love the high ceilings. It looks like a lab that came out of like a sci-fi movie or something. I think it's such a cool design. The windows are so big. My bench is on the far right. The windows facing the botanic garden, so I can see the trees change throughout the season. Good morning. I am going to collect my leaves from the greenhouse. I'll be scanning them first, then I'll be taking pictures of the leaf surface. I had a bit of an accident last week. This was an experiment I had to repeat because I lost all my data, which was on the USB stick, which went corrupt. I used to save my files directly to the USB stick, which worked until all my files had vanished. So yeah, I really don't trust USB sticks. I mean, it's also my fault that I didn't save all the images to the original computer, which I'm doing now because I don't want to repeat things again. Now I am taking pictures of the leaves on the microscope to use them later for some data analysis. Also on Tuesday mornings every week there's a seminar which involves several groups that are studying plant microbial interactions in the roots. This is my favorite seminar series because they're talking about things that I love. It's just so fascinating to hear what other people are doing. It would be such a dream to be able to present in that seminar one day. Anyways, my day went on taking images on the microscope. Took pretty much most of the morning. I finished just before noon, so I did some washing of my membrane and did my secondary antibody for my Western blot. Left that over lunch. Lunch was so nice. It was so warm today. The sun was out, the sky was blue, so we were having lunch outside. In the afternoon, I was trying to extract some DNA. This was the bacteria that I grew yesterday. First, I'm making a stock and keep the bacteria frozen. It's on.
The one that my god. Nice.
Well, the 3 bands. Yeah.
So it's mean that
Yeah.
Anyway, and the membrane is very nice.
It's white.
It's white. That's an improvement, even if
I mostly just did more protein gels and Western blot. My supervisor was there for imaging one of the membrane. He was quite happy about what we got, which was nice. I continued with my DNA extraction. I spin the bacteria. We spin them, meaning we put tubes in this machine which spins at a ridiculous speed, literally like 1,000 times per minute. And of course, they get all dizzy and pass out. So then they collapse to the bottom of the tube. And then I treat all these bacteria with a cocktail of harsh chemicals which break them open like piñatas and spill everything inside them, which includes their DNA, which is what I need. Yeah, it sounds quite brutal, but that's how we collect their DNA. So that's what I was doing. So why am I doing plant sciences? Because plants are important sources of food like rice, wheat, vegetables and fruits. In my head, plants equals food, which equals health. With the limited amount of resources, I think we need to figure out better ways to grow food. Initially, I wanted to become a plant engineer to make plants that can grow better in harsh conditions like droughts, very hot climate and tolerant to diseases. I've always been telling people around me that I wanted to work on plants, especially on food crops, but my undergrad was not really related to plant science. I finally got into a master's course that's even called crop science, and yet my interests keep on jumping. So you know what? I still haven't gotten around to working on food crops. I'm mostly using model plants in animal research, for example, where you're testing different drugs. People would test it on mice because you obviously can't test them on humans. In plant research as well, there are several plants that are only used in research. They don't produce edible parts. You use them more to understand the plant itself. So yeah, I've been mostly using models, which is still cool, but yeah, I still have a long way to go until I can finally work on crops. Morning. I just got to the lab. I'm just going to the greenhouse to check a few plants. This morning I have a meeting. Every Wednesday morning students have a meeting with the supervisor. I won't be doing any lab work, and in the afternoon I'll be finishing off my Western blot. Hopefully get some results before the presentation tomorrow. In my current lab group, we have a lab meeting every week. We basically have to present roughly about once in 3 weeks. At first, I thought that was quite a lot, but after a while, I got used to it. It's a really nice opportunity to get more comfortable speaking in front of the group and presenting whatever I've been doing, even though I'm not getting the data I want to present. I much prefer doing presentations than writing. I never really focused too much on visual communication until this one incident in my undergrad. We were told to make a poster as part of my final year project, and I made an awful poster. It had lots of text, one figure showing some microscope images, but obviously it wasn't helping. When I presented my poster, my examiners just trashed my poster for like straight 10 minutes, which totally made sense. It was such a disaster. I got such a low mark for it, and I really needed a revenge. So from there, I just tried to convert everything into a diagram, and at the end of the project there was the viva, which is like an oral presentation of my project. This time I put tons of diagrams. At the end, my examiners were not so disappointed anymore. But since then, I genuinely started enjoying making presentations. Finished eating dinner. I need to finish off my presentation for tomorrow. That's what I'll be doing this evening. I also have a poster that's due in 2 weeks and I haven't started working on it. Let's see how that one goes. I'm still not hearing back from the internship that I applied for. I need to start applying for jobs. It's just so nerve-wracking to wait. I just keep on checking my emails. It's not nice. A lot of my friends got into their PhDs. They got into their first choices. I'm really happy for them. It's also really inspiring to have so many people around me who are so passionate about what they're doing. Even though things haven't worked out for me this year, seeing people around me get what they want to do really inspires me. It makes me want to stay strong. But yeah, for now it's just waiting and applying to more things, which I haven't started yet, but I probably need to soon. It's already May. Anyways, see you tomorrow. Good morning, it's Thursday. I have a presentation to do in the morning. I have like 1 hour, so hopefully I can finish up the last bits. I don't have anything planned today in terms of experiments, so I think I'll just do some data analysis. I finished my presentation. It was alright, and I ended up having a discussion with one of the postdocs about one of the experiments that I was doing and having problems with. I'm really lucky to be in a team where everyone is so supportive. They always help me out. After that, I came home. Today I made Japanese curry. It's one of my go-to meals when I don't want to do much. It's the only Japanese food that I can cook. It's super easy. You just chop up some onions, carrots, potatoes and meat, mix them with the Japanese curry roux. You can find in Asian stores. I've seen them in Sainsbury's and Tesco's as well. You cook some rice together and then that's that. I go for speed whenever I cook. I don't follow recipes, but it works and it tastes good, so that's all that matters. I usually make 3 to 4 portions per meal. I don't like freezing stuff. I just eat the same thing until I finish. Okay, live reaction. Just now I received an offer to work in one of the labs that I applied to. Oh, I'm so glad. I'm so glad it worked out. I can't believe it. Oh my gosh, I was immediately drawn to his project. I just thought it would be such a dream to be able to do something like that. I practiced my interview presentation in front of my current lab group and they gave me some really solid feedback which helped me prepare better for the interview. This also means that I need to move out of this house to a new city. Personally, the current house that I'm living in is a place that I've never felt more at home than anywhere else that I lived in so far. That's one of the things that's a bit sad, but this opens up so many new doors. Because it's a 1-year internship as well, it means that I can reapply, basically try again. I thought about just giving up science. I'm not good enough. It's something I really enjoy doing. It is quite challenging at times, but I think it's very rewarding when something comes out of it, and there are things that I want to explore more. I'm really thankful that the new supervisor has given me this opportunity. After this internship, hopefully I can get into a PhD. That was once my dream. I'm really excited, quite nervous, but yeah. Good morning, it's Friday. I'm on my way to the lab. I got myself a pastry to celebrate and hopefully finish things off before the weekend. I love the weather this week, it's been great. Friday was pretty chill. I did some PCRs to get some DNA fragments which I needed for cloning, and I also transferred some of my plants to a new plate so that they can grow well. Since I wear headphones for most of the day, I go through so much music every single day. I'm a big fan of classical music. I usually put on the BBC Radio 3 when I'm having dinner, and from time to time I find music that I really like. I add that to my playlist. I recently discovered the Brahms Symphony 2 and 3. I only knew Symphony 1 and 4. I've listened to it multiple times, but I never really got into Symphony 2 and 3 until I had some snippets somewhere, and I've been playing it non-stop. Now I've listened to several different recordings of it. I really like noticing the subtle differences in the way different orchestras play. I wish I knew more about it. I've also recently made a new playlist called Soundtracks because I like background music in animes and films. Some of the animes like Your Lie in April, Garden of Words, Your Name also has good soundtracks. I also added some from films like Interstellar, Theory of Everything, which is a film that I also really like. I have one playlist for symphonies, and I have another playlist for all the piano pieces like etudes and ballades. I have another playlist for concertos and anything that has a solo and an orchestra, so like Carmen Fantasies, Theme of Paganini. Recently I also tried something new, which is to listen to classical music in shuffle mode. Actually, shuffling classical music is also quite fun. You go from one movement of one symphony to another movement of another symphony, and sometimes it just works. But yeah, I wonder what other people are listening to. Cutting my gel. I'm wearing these fancy glasses that can help me see DNA near UV light. I'm extracting the DNA band that I need for later. Here I'm trying to measure the amount of DNA in my tube. Now I can use them for other things. Friday, finally. I just came out of the lab a bit early. I'm gonna do a bit of grocery shopping, go home and chill. It's such a nice day. It'll be so nice to read a book outside now that the days are quite long. Nice to be able to go out through the botanic garden every day. Such a beautiful place. I just squished a bug on my head, I'm sorry. Hope you enjoyed watching my week. It was quite spontaneous, but actually I had a lot of fun filming this. Hope you have a nice day and see you soon. Bye! Surprise, surprise! I'm skating! I finally took out my skates. The last time I wore my skates was when I was in Japan in December when I released my last video. It's been a while. I was heading to my colleague's PhD graduation party. Congrats to him. It was a beautiful garden, there were pizzas and it was really fun. It felt so nice to skate again. Meant to be happening somewhere around here. I'm not sure where I am at the moment. It's the first time skating and the roads are so bad. I'm so scared of skating here. Hopefully I find some people that I know. Yep, indeed, I was in the wrong place. I am gonna skate back to the main road and hopefully find the right place. The cows. What? No way, that's so cute.